Today we'll be starting with a new chapter, chapter 5 in the textbook, that is titled Enzymes. And we are going to look at enzymes with the sense of them being biological catalysts. So, first of all, we need to talk about what a catalyst is, right? And even before that, as an introduction, right? All enzymes are proteins, which means, right, they are made up of amino acids, and they get that right. They are coded for by the DNA, right? Because that's what's right. How proteins are made in the body. They are obviously made from amino acids, and they are coded for by the DNA as and when required. And all enzymes are proteins. And that is important because when we talk about certain properties of enzymes, right, they will be based on the fact that they are proteins. Now, right, these guys are biological catalysts, as I mentioned, right? So first, let's define what a catalyst is. So a catalyst is a substance that is involved, well, I shouldn't say involved, a substance that increases the rate of a chemical reaction and is not changed by the reaction, right? So it's something that helps the reaction to take place faster or more efficiently. Uh, Obviously, it has an effect on the parameters, as you could say, of a reaction. That is why it makes it more efficient or makes it go faster. But it's not involved in the reaction in the sense that it's not taking part in the reaction itself. So there's nothing changing, right? And basically how that happens in many cases is, right, the catalyst brings the reactants close together for the reaction to occur. So instead of relying on random collisions as they would, we've talked about diffusion and all that. So catalysts are not waiting for, two molecules of a substance of different substances to come into contact by random collision, right? They're going to bring those two together. However, right, collisions are how enzymes work as well. So we get into that, right? But this make it more efficient. So that is a catalyst. And then enzymes are biological catalysts. Why are they right? So they perform the function of a catalyst, but they're biological because they are present in living systems, right? So it's not just humans, it's all living systems. They have uh, enzymes. So enzymes are proteins, right? They function as biological catalysts, and what they do is that they are involved in all metabolic processes, right? And metabolic means, right, a sum of bond breaking and bond making, right? Which basically means bond breaking is when larger molecules are broken into smaller ones, and bond making obviously is when smaller molecules such as the building of polymers from monomers, right, or even the building of lipids from a glycerol molecule and fatty acids. So when smaller molecules are built into larger ones. Now, there's a specific term for each of these, right? And those are anabolic and catabolic reactions.
Like all enzymes are proteins, but not all proteins are enzymes.
Oh,
You
no,
know?
no. Right? I mean, yes, your understanding is correct. So all proteins are not enzymes,
Okay.
right?
And can you define that? What is a metabolism?
Right. So as right, metabolism is the sum of all the reactions that involve the breaking of bonds in the body, such as digestion,
Yeah.
right? And digestion is an example. And all the reactions that involve the making of bonds, so making larger molecules from smaller ones, such as making proteins themselves, right? Or making glycogen, or right, and any other, right, the process of DNA replication where nucleotides are brought in, and the entire molecule is made, DNA or RNA. So all of those are bond making. And so the term for bond, the reactions which have bonds being broken are catabolic reactions, and the ones that involve bond making are anabolic reactions. So if you look at them individually, they have their own names. Anabolic is for bond making, and catabolic is for bond breaking. One way I try to remember this, although I do sometimes get confused myself, right, and I'm not being particular, right? Catabolic, you can either look at it this way, that C for construction, but it's
Yeah.
the opposite. It's not construction, it's breaking down. Or catabolic, you can look at it as, oh, it's bond breaking, so catabolic catastrophe, right? And so that's bond breaking. And the other one's obviously anabolic bond making. I haven't, right, thought of a mnemonic or something like that for this, but if that helps you remember, uh, right. And so the sum of these,
Like, can you please tell the examples of anabolic reaction? 'Cause-
anabolic reaction is making proteins, or making, as I mentioned, glycogen or DNA replication, right? So making DNA from nucleotides. So we talked about nucleotides in the previous chapter, right, as a biological molecule. Or making proteins from amino acids. So these are all bond making. So you're putting together amino acids to make proteins, you're putting together nucleotide bases to make DNA, you're putting together glucose molecules to make glycogen, right? You are making phospholipids for the cell membrane. I mean, phospholipids isn't part of your syllabus, so uh, right, or any other reaction in which a bond is being made. So for example, you are making ATP from ADP, right? And so that is, right, a bond making reaction. Whereas bond breaking, right, is simply put digestion. So all those complex molecules such as starch and proteins and lipids, they're being broken down into the individual sugar units or the individual amino acids, or into the glycerol and fatty acid molecules, or right, when for example, during, if a cell is dying, then it is broken down, or during immune reaction, when a phagocyte takes up a bacterial cell and digests it. So in all those cases, uh, right, bonds are being broken, and so those are catabolic reactions.
And like enzymes are just there to fasten up the process.
Yes, and right, they take part. So in an biological system, in a living system, enzymes are involved in pretty much all reactions, right? So
Even in a single cell.
yes, all metabolic reactions that are occurring everywhere in the body involve enzymes. And again, metabolic reactions in terms of bond making or breaking, right? So for example, active transport is a process. It is not a metabolic process. It is just the movement of molecules of a substance from a region of low concentration to a region of high concentration, right? So enzymes are not involved in that case. Proteins are, as you talked about, right? So they're carrier proteins that do that. But again, not all proteins are enzymes. There are different kinds of proteins. Does that make sense?
Yeah.
Okay. So again, enzymes are involved in metabolic, right? When there are reactions taking place, right?
Okay.
So in a living system, in a biological system, enzymes are involved in all metabolic processes. And the thing about that is, for example, right, the, the question is, can certain reactions that occur with the help of enzymes occur without the help of enzymes? And the answer is yes, right? So, for example, starch that's present in potatoes or rice or wheat, so the bread that we eat, right? Starch, if you put it in water, and it will break up into maltose, which is the disaccharide, and perhaps eventually into glucose. But this will take multiple hours or days, or it will be so slow that it may take years to happen on its own, right? So it's going to take a very long time. So long that the living organism will no longer be living, right? So we can't wait for hours or days or even months and years for a single molecule of starch to be broken down into glucose, so we finally get that energy. It doesn't, we can't survive. And so the thing is that living organisms, right, need these reactions to happen very quickly, happen or occur very quickly. Why? To be able to sustain life. So if you eat food and it's just sitting there in your stomach for days, because it's slow, you're waiting for it to happen on your own. Your cells need that energy. They need that glucose to respire, and all the other processes. So they can't wait. They're going to die, right? And when cells die, you die. I mean, the organism dies. right? On a mass scale, obviously. So, uh, yes, enzymes are required, right? And this, right, the thing about enzymes is one, you do not need a lot of them. So, right, very small quantities of enzymes can make a huge difference. And the rate at, by which, right, different enzyme speed, are work at different speeds, have different rates of enzyme activity. So, for example, there is an enzyme called catalase, right? And this enzyme, what it does, it is commonly found in cells throughout the body. And what it does is it converts hydrogen peroxide to water and oxygen. Because hydrogen peroxide, what is hydrogen peroxide? Hydrogen peroxide is H2O2, and this is toxic to the body, right? And so what it does is it converts it to H2O plus O2. So if you want to balance this, that's how it's going to be, I believe, right? And so this is an enzyme catalyzed reaction. And the speed at which it does, right, it can catalyze, if I'm not mistaken, 4,000 molecules of hydrogen peroxide per second. In 1 second, it can convert 4,000 molecules of hydrogen peroxide into, uh, you know, water and, sorry, not 4,000, 40,000, right? My bad. I thought that the number is a bit low, right? For an enzyme, it's 40,000. So, correction. So, 40,000 molecules of hydrogen peroxide are converted by this enzyme per second. That's how fast the enzyme catalyzed reactions are. Again, not all of them are as fast, but right, even the slower ones, so to speak, are increase the rate of reaction by many fold. Any questions?
No.
Okay. Now, right, enzymes are specific. So enzyme specificity. Enzymes are specific as to which substrate. I'm sorry, if you could mute your mic if you don't have a question. I'm sorry.
I'm
Thank
sorry.
you. Yeah. Enzymes are specific as to which substrate uh, binds to them. So, right, the substance that binds to the enzyme is called the substrate. So, substance that binds to enzyme, and then that is converted to the product. Now, keep in mind, right? It can go both ways. It can be bond breaking and bond making, right? So you can possibly have two substrates forming one product, or you can have one substrate being broken down into two or more, right? So I'm just, two, it's not, it could be more than two, right? But point is, 2 to 1 is bond making, 1 to 2 is bond breaking. So this is bond making, which is anabolic, and this is
Is the
catabolic.
first one the bond breaking?
This one,
Yeah.
this is bond making.
How?
Hmm?
Oh yeah, yeah. Sorry.
You are taking two molecules and joining them to make a larger molecule. So that is bond making, which is anabolic.
Yeah.
Okay, now how this works is, right, enzymes are proteins, and so they are folded into specific shapes. And then there's a part on the enzyme molecule that is called the active site. And it is the active site that, where the substrate binds. Right. So there's a part on the enzyme called the active site, and this is the binding site for the substrate. And the thing is that the shape of the active site, it is specific to its substrate. So it looks something like this, right? And as you know, how you know good of an illustrator I am, if you have, right, an enzyme that looks like this, like Pac-Man. And so seeing the shape of the active site, the substrate will probably be shaped like something like that, right? So it's able to fit in. So even if there's like a slight difference in the shape, for example, if you have a substrate that is like this, and so it's similar, but you can see that, right, this end is not pointy, it's sort of curved, and so this will not be able to fit in, right? There are cases where similarly shaped substances are able to fit in, and whatever, but that is beyond the scope of your syllabus. So, right, let's just say that they're very specific, and so only the substrate that matches the shape of the active site, that's able to fit in properly, will be processed and converted to the product. Again, the product can be of an anabolic or catabolic reaction, so that depends on what kind of reaction is being catalyzed, right? So that is specificity to, right, the substrate in one sense. And then the other thing is, right, uh, they are specific to the kind of reaction that they are, right? So some enzymes are able to catalyze more than one reaction. Some, but for the most part, what we need to know is that they are specific in their reactions as well. might catalyze only one reaction. What that means is, for example, if you've got starch and that's being broken down to maltose. Sorry, just a minute.